Okay, it's very difficult to actually to frame work which took more than 20 years since I'm working in Messina and in 15 minutes, but I will try to be very fast and synthetic. First of all, just taking a nice view of our institution, which is located just on the sea in the Strait of Messina, and it's not far. Can I make, yeah? Can you see the air? No, okay. I will then use pointer. It's not far from the region, which you can see, oops. Okay, we can start. Can I use my pointer maybe? Yeah, but anyway, we are located here, and here we have very peculiar area of Mediterranean Sea, which is actually Mediterranean ridge, and with very high tectonic activity, which is actually reason to form very peculiar hydrological formation named deep sea hypersaline anoxic lakes, which are located on the seabed of Mediterranean Sea, and they look like somehow like this. Then you see really normal seabed, which is, I would say, around 3,700 meters below sea level. And first such formations were discovered in 1983, and then since couple of, I would say, almost 40 years, we continue to work on these peculiar ecosystems, and we actually published last lake we found, Lake Hephaestus, in 2019, and we bring, because before our studies there were 5 lakes known, we actually bring this number to 9. And just can you imagine that in 21st century you can discover new hydrological formation on Earth, which is less studied, I would say deep sea habitats, less studied than Mars surface or the moon surface. And you can find even formation with the dimension like, for example, Medea, which is over 45 kilometers length and more than 7 kilometers wide, and some of the lakes, they are 300 meters or 400 meters deep. It's really huge formation, and still, I don't know how many they are still to be discovered. Okay, I will just make some, I would say the trip. Just imagine you are on the ship and you are discovering new lake, absolutely new ecosystem, absolutely unknown hydrochemical and environmental setting. We will start. First, what you can see by echo sounding, you will see the very horizontal line which are actually separating lake from the seawater due to increasement of density of the lakes, because they have all of them, they have the different value, but almost all of them, they are at the level of salt saturation. And salt can be sodium chloride or even magnesium chloride. In case of, for example, this lake here, they are depicted by red. It's Discovery, Hephaestus, and Cryos. All of them, they are filled by magnesium chloride, and they are anathema of life. That means we are just breaking the paradigm of the biology where you will find liquid water, there is life. No, there is no life in these lakes. And they look like very interesting because all of them, as I told you, separated due to increase of density, and they are relatively stable, and due to absence of circulation, all of them anoxic. There is no oxygen. They are salt, 300 atmosphere or even higher the pressure. Sometimes I would say concentration of the H2S, it's 3 or 4 times more than lethal dose for the human beings. They are really sulfidic. Okay, and another picture actually I prepared especially for Christian, who is present here, because if you will start to look for dark primary production, they are really, for example, this is the column over the Medea lake. And these green bars, it's values of dark primary production, or in this case, dissolved inorganic carbon fixation. And you can see on the interface between seawater and basin, hypersaline basin, it's really bloom and increase of the fixation. That means it's like a hotspot of not only biodiversity, but it's hotspot of activity, metabolic activity, which can be like a key or pivotal role for the deep sea environment. And yeah, we made then also the transect for the whole Mediterranean Sea, and actually this is the rule. You are approaching the anoxic basins, and on the bottom you see the peak of activity of carbon fixation. This means dark primary production. Without life, you have the organic carbon there produced in situ. And can we try? Yeah. And then go to make trip. It started? No. Yeah? Ah, okay. Yeah, because here's standing. Okay, this is actually, I got this video from our colleague, collaborator, Virginia Edgecombe from Woods Hole, and this is, I think I can, because I also would like to see, because my screen here is stable. Can we switch off the light here? Can we, no? Okay. Because it's really some fascinating stuff. For example, you cannot see here, but this seems to be skull of the dolphin here. Because as you might imagine, hypersaline anoxic stuff, it's somehow, I would say, controversial for the life. And even alive animals in these depths, if they reach the lake, they are simply dying. And what I actually, the movie is relatively long. I would just skip to, can I go here? No, okay. No, I cannot even control this movie. Can we jump to the 8 minute? Yeah, and as I said, you are really like, you're on a helicopter flying over the lake, but just forget then we are 3,500 meters deep. It's really amazing. It's like a beach you see here. No, maybe I will skip because I have just, okay. And this is what I was speaking about the separation between the lake and seawater due to increasement of density of the brine. And as you can see this is the interface, and this manipulator is activating the push core to take the samples. And as you can see, the interface became again, I would say, stable, and again separating lake from seawater. It's really somehow impressive. Okay, thank you. And just, we can jump to another movie. And it's again, just example, not of Italian science, because we don't have ROV like Jason. It's something like 13 operators and more than, I would say, $1 million per day or $500,000, something incredible. And this is another one. This courtesy from Dr. Andrea Antunos. Actually, they made this movie in anoxic lakes in Red Sea, and they are not so deep like Mediterranean one. This is the depth of the lake. And they were actually specially constructing the floating ROV on the interface to see how beautiful and how densely inhabited this peculiar ecosystem. And we are moving, look, boom, we're entering in the interface. Here the interface. Now the buoyancy will be stabilized. For a couple of seconds you will see, okay, we have some zooplankton. Very beautiful. Now it's stabilizing the ROV because of the calculation between the density of seawater and the brine. And they are evaluating position. And now we're moving. This is interface. Color seems to be due to very peculiar microorganisms called halarchaea. And they are, at least on the surface, they are red, intensively red. And actually, that's why you can, all this super saline, natural, or even solar salt ponds, they have this reddish, typical reddish color due to halarchaea present there. And also, if you be very attentive, you see multiple zooplankton living there because they are really predating this huge amount of biomass concentrated on the level of, I would say, a few centimeters. Okay, thank you, because I don't have too much money, no, time, sorry. Okay, then let us go, and actually what can be created, it's almost digital stuff. Innovative, of course, because we don't have ROV. We don't have ROV like KAUST Institution or KAUST Academy of Science, King Abdullah, and we have brain. And then we created some technique to sample the interface without perturbing during the sampling. And actually it's relatively simple, but I would say it's require at least nice weather without shaking and 3-4 meters high waves. And also, actually you need very, very, I would say, sophisticated team on board of the specialists and engineering techniques, because you have to go very slowly to the interface, and during the penetrating the lake, you are registering the actual position of the interface. Then this interface, of course, disturbed, and perturbance here is very high. That's why you go deeper in the lake and then move away, at least 50, 100 meters away. And then knowing already the positioning of the seawater interface gradient, you are just moving upwards very, very slowly and collecting samples at the depths you need. And actually our record was a resolution about 10-20 centimeters resolution to sample the interface on the depths of 3,500 meters blindly. You are blind because you cannot see anything. You just have the one sensor, its conductivity, to see the salinity. Okay, then you collect the samples and start to analyze typical microbiological, molecular biological part of the work. And what I can see, what I can show you here, we just place the environmental setting, which you can find in the interface in brine, with the all known extremophilic isolates. Known so far, I think I took this from 2013 paper, but it didn't change much. And you can see all our environmental settings containing alive microorganisms. They are far away from the known microorganisms. This means we have new microbiota there. And indeed, this is now actual tree of life with very, very new phylum level bacteria and archaea. And actually many of them, they are present in these deep hypersaline lakes as the active, I would say, playing pivotal role in these environments. taxa. They can be all measured only by activity, metabolical activity, to understand actually they are active there or they are present there as the dormant or sleeping cells. And that's why you have to go to study these microorganisms by finding some mRNA transcript, or in many cases also proteomics as well. Then you are analyzing diversity and finding the more, I would say, principal organisms. In this case, I just highlighted them in red. And we have acetothermia, we have methanohalophilus, MSBL1 candidate division as well. And then we have these two groups, which is nanohalarchaea and halarchaea. Then try to cultivate them and try to understand the metabolic capacity. And it's coming out also following the literature from colleagues. You can understand, or you can put your vision and create the model about the metabolism of these pivotal organisms. Going ahead, genomic, actually decodification, or trying to understand how they work using genome sequencing and annotation. Then creating the actually models how they can be functional in the extreme environments. In this case, we try to create new phenotypes of halarchaea, which is sulfidogenic anaerobic organisms, never found before our studies. Then trying to insert them in the model to actually try to somehow understand how these lakes, or even the interface of the lake, can be functional, creating and actually involving all the participants in this kind of cycling of nutrients and carbon. And then it just be small gallery show about new life or new type of life we discovered recently. It's nanohalarchaea. It's some kind of organisms which are living as ectosymbionts on the normal prokaryotic bacteria or archaea as a, I would say, not really 100% parasitic, but in some case they have even mutualistic relations between host and the organisms. And as you can see, this is the normal halarchaea, and you have the small spots here, bright spots, and this is nanohalarchaea, which have dimension not more than 500 nanometers, very small genome, and they look like this. For example, this is nanohalarchaea sitting on the, we call it in laboratory on mama, because this is the host, actually normal halarchaea, and this is nanohalarchaea. As you can see, pretty happy both of them. Then trying to make some gallery, to make new names. We have now new candidate Nanohalobium constans, which is actually first representative of the new class Nanohalobia, and they look like this, using Photoshop, of course. And that's all, yeah. Then start to make model, and actually they are small, but they anyway target of the nanohaloviruses. And here, for example, the nanoarchaea, you can see the bar, which is 200 nanometers, and they are full of viruses. And also they have some enigmatic, I don't know what it is, still we have to discover, but these tubular structures, which we found here and visualized by cryomicroscopy, they are very similar to known filamentous viruses from in this case it's thermophilic archaea, but the structure somehow similar. This means the world is really amazingly diverse, interesting, and can be digitalized as well. Thank you for the attention. And this is my team, and thank to all my colleagues. And my members of our group, and yeah, thank you very much.